Hello everyone. So we will discuss start today today's session. So in this session we'll basically discuss about the process and mechanism of B cell activation and B cell then maturation and differentiation of the B cell. Right, as we know from a very uh, brief in a very brief way that when a foreign antigen comes to our body, and then the this um, lymphatic cells, like for example B cell, T cell, they has to be activated. And so the process we have discussed till now, that how T cell gets activated, and how T cell in turn activates the B cell, right? So that thing we have discussed till now. So in this session we will see how B cells can upon one antigenic stimulation, how B cells get activated, and what is the mechanism of the signal transduction, how do they proliferate, and how do they actually help to combat the uh, infection. So that we will see in this session. So we will first start with B cell maturation. This is a very brief process of B cell maturation. Uh, just a minute, let me change the pointer to the layer pointer. So in bone marrow, as you know, B stands for the bone marrow. So in the bone marrow, the progenitor B cell it undergoes Ig gene rearrangement, which is immunoglobulin gene rearrangements, to form a mature B cell, right? And for a progenitor B cell, the marker is B220 or CD45 bar. This is the surface marker for a progenitor B cell. So after the Ig gene rearrangement, it will start expressing the immunoglobulin genes in the surface, and it will be a mature B cell. So that entire process we will see how this gene rearrangement takes place. Then from bone marrow it will come to the peripheral lymphoid organs. So here it will come, and when until it is encountering an antigen, those B cells will be called as knife B cell. But when they will encounter an antigen, then these B cells will be classified, that which B cells will stay alive and which B cells will die. So when this plus antigen and Th cell, because as you know from the previous sessions that Th cell, helper T cell, has a role upon antigenic stimulation. This helper T cells gets activated and it activates the B cell, right? So this antigen and Th cell activates the B cell. So the knife B cell will be converted into the activated B cells, and the B cells which will not encounter antigen, those B cells will be negatively selected, they will be died, right? And then this activated B cell will undergo either affinity maturation or and the class switching. So in affinity maturation uh, and the class switching process, it will be divided into two kinds of B cell. One is memory B cell, one is plasma B cell. Plasma B cell will secrete antibody, memory B cell will not secrete, but it will keep the information of the antibody for this particular antigen. So when this same antigen will infect the second time, then this memory B cell will show its activity, and it will elicit a quick response against this antigen. In contrast, the plasma B cell will secrete the antibody upon getting a antigenic stimulation. So this is a very brief way of uh, describing the maturation process of B cell. So let me know if you have any doubt. So after that we will move to the maturation of the pro B cell to pre B cell. So as you know that the B cells, from this figure we can know that the B cells, uh, the maturation process of the B cell, it occurs in the bone marrow. So in bone marrow, this is a bone marrow stromal cell. So the B cells are attached to the bone marrow stromal cell by interaction of VLA4 and VCAM1. VCAM1 is expressed in the bone marrow stromal cell, and VLA4 is expressed in the pro B cell, right? So through this VLA4 VCAM1 interaction, these pro B cells will start expressing a surface receptor which is called c-kit. This c-kit on the pro B cell that binds to the stem cell factor SCF on the stromal cell. This is the SCF on the stromal cell, which is stem cell factor that binds with the c-kit of pro B cell, and which triggers a signal mediated by the tyrosine kinase activity of c-kit. C-kit has a tyrosine kinase activity, and by this activity it will stimulate the pro B cell to express receptor for IL-7. And from the bone marrow stromal cells, IL-7 will be secreted and it will be received by the IL-7 receptor in the B cell, right? So upon receiving the IL-7 by the IL-7 receptor of B cell, these B cells will start proliferation and differentiation. So it will be proliferated to, so after this IL-7 binding, it will be converted into the pre B cell from pro B cell, and from pre B cell, when it will be proliferated and differentiated, it will form immature B cell. So this is the process of. So here you can see that from in bone marrow, the pro B cell will be converted into mature B cell. So yet till now the B cells are not matured. So first the pro B cells will attach to the bone marrow with this VLA4 VCAM1 interaction, which will enable the c-kit protein surface receptor of pro B cell to interact with the stem cell factor SCF on the bone marrow stromal cell. So this interaction triggers a signal where stimulate the pro B cell to express the IL-7 receptor. And now this bone marrow stromal cell will secrete the IL-7, which will be received by the pro B cell, and this IL-7 and IL-7 receptor interaction, upon this signal binding, the B cells will start proliferating and differentiating. So it will give rise the pre B cell. From pre B cell it will give rise to immature B cell. So that's how the pro B cells are converted into immature B cell in bone marrow. So any question you have, you can ask. So this was the process for maturation of pro B cell to pre B cell. Then we will see the stages of B cell maturation in detail in bone marrow. So in bone marrow, there are two phases. One is, so in bone marrow it is antigen independent phase, and in periphery it is antigen dependent phase. So in bone marrow, what's happening? Lymphoid stem cell will, which we actually saw here, this Ig gene rearrangement. So here in this case you can see that the progenitor B cell after this gene rearrangement can be converted into the mature B cell in the bone marrow itself, and this is the process of proliferation to make immature B cell from pro B cell. And how this gene rearrangement, if we will see, then first the lymphoid stem cell will undergo DJ recombination in the heavy chain DJH, which will make Ig alpha and Ig beta heterodimer of BCR B cell receptor. then there will be a VDJ recombination where pro B cell will be converted into the pre B cell, where along with the BCR Ig alpha Ig beta, it will have the heavy chain as well as surrogate light chain of the pre BCR, and then the light chain will be rearranged, means this variable region or heavy chain region is already rearranged, but the light chain here was surrogate. So after the VLJL recombination, the light chain will be light chain gene will be will undergo recombination. So it will make the IgM and Ig alpha Ig beta heterodimer, that which is the immature B cell which we saw here, immature B cell. So again, from lymphoid stem cell, DJ of heavy chain recombination will give you the pro B cell which has Ig alpha Ig beta heterodimer. Then VDJ recombination will make pre B cell. In this pre B cell there will be Ig alpha Ig beta heterodimer, heavy chain, and the surrogate light chain, that is the pre BCR. So upon this VLJL recombination in the light chain, the surrogate light chain will be converted into the permanent light chain, and that will give you the IgM here, heavy chain as well as light chain, along with the Ig alpha and Ig beta. That is your immature B cell. Now so this is the antigen independent phase in bone marrow. There is no involvement of any antigen. After that, when it will come to the periphery, then in periphery there is antigen dependent phase. So in the periphery, the immature B cells will be converted into naive B cell, right? Naive B cell, and from upon antigen stimulation, this naive B cell will be converted into the mature B cell, as we saw here. When it will come to the peripheral lymphoid organ, then it will start encountering antigen, and then antigen will make this naive B cell, antigen and of course activated helper T cell. Both of them will make this naive B cell to activated B cell. So when it will come to the peripheral lymphoid organ, this naive B cell will be converted into mature B cell after antigen stimulation and activation by helper T cell, and it will also start expressing the IgT. So these two are the surface immunoglobulin. They will be expressed at first for a surface of a B cell. So that is your mature B cell. So these are the stages of B cell maturation in bone marrow, and of course also in periphery. So in bone marrow it is antigen independent, in peripheral lymphoid organ it is antigen dependent. Now if we see the sequence of expression of membrane immunoglobulin, then you can see that in pro B cell pro B cell contains only Ig alpha and Ig beta. Then as we saw here, the DH recombination, first this DJ recombination will give you the Ig alpha Ig beta heterodimer, that is your pro B cell, right? Pro B cell. From lymphoid stem cell you are getting pro B cell which you can express Ig alpha Ig beta. Then upon this VDJ recombination you will get a pre B cell where Ig alpha Ig beta heterodimer as well as the heavy chain will be there and a surrogate light chain. So is pro-V-cell Ig alpha Ig beta. Then in pre-V-cell, after this VDJ recombination in the H chain, and you will see there is Ig alpha Ig beta was previously there. Then there is this VDJ. After this VDJ recombination of the heavy chain, you will get a heavy chain, and you are getting a surrogate light chain, which are lambda 5 and V pre beta, B pre B. This is the surrogate light chain. So, which can cross-link by stromal cell ligand, that will stop this VDJ allelic exclusion. So after that, from pro to pre-V cell, the next step is immature V cell, that is occurred by VLJ recombination. So now this light chain will be light chain gene will be rearranged to make this VLJ rearrangement to get a immature B cell, which along with the heavy chain it can have a mature light chain, which is either kappa or lambda, along with the Ig alpha Ig beta heterodimer, right? So that is the process of conversion of a pro B cell to immature B cell, right? So this is the sequential expression of membrane immunoglobulin. So, and this is the in the terms of gene rearrangement, which can actually convert this pro B cell to immature B cell. So if you have any doubt, please let me know. So this is a sequential expression of membrane immunoglobulin gene, which actually which actually the which actually is showing that the how this expression of immunoglobulin genes are uh, converting the pro B cell to mature B cell. So now the signals for B cell activation, because now till now we saw that in bone marrow the B cells undergoes the gene rearrangement, and from the lymphoid stem cell to pro B cell, and it will finally convert it into immature B cell. Right. So now this immature B cell has to encounter antigen in the lymphoid organ, and it will convert it into the mature B cell. So how this activation does occur? So it is by binding of a type 1. So this can be occurred by two ways. One, when when the binding of a type 1 thymus independent antigen to B cell, and the second option is binding of a thymus dependent antigen. Right. So when the thymus independent antigen binds to the B cell, so now after this, when this thymus independent antigens bind and to directly to the BCR of B cell, here you can see this is the antigen and this is your BCR. So after this, upon this binding, the two signals are provided, right? And in the second case, where it is the case of thymus dependent antigen, then the thymus dependent antigen provides signal 1 by cross-linking with membrane IG here, right? And the second signal comes when the CD40 of B cell is interacting with CD40L of helper T cell. So this provides the signal 2, and these two signals provide the means for activation of B cell. So for thymus independent antigen, there is no cross-linking; it will bind to the BCR, right, to the membrane immunoglobulin, and which is enough to provide both signals. And in the case of thymus dependent antigen, the antigen will cross-link with the membrane immunoglobulin. This will give you signal 1, and this CD40 of B cell and CD40L of helper T cell, this interaction will give you the signal 2 that will actually help to activate the B cell. So that's how the B cells, the immature B cells can be this naive immature B cell that can be converted into the mature B cell in the peripheral lymphoid organ when it will interact with the antigen. So that's how it can be converted into the, from naive B cell it will be converted into the mature B cell, right? So now we will see the signal transduction by BCR, how this B cell receptor of BCR that will provide these two signals, how these signals get transduced, that we will see. So so firstly, the antigen will come, it will bind to the membrane immunoglobulin variable region, and then it is associated with the Ig alpha Ig beta heterodimer, which is also a part of BCR, that has a ITAM motif, which is immunoreceptor tyrosine based activation motif, that is rich of tyrosine residues, So this ITM region of Ig alpha Ig beta, it interacts with several members of the SRC family of tyrosine kinase, for example FYN, BLK, LCK, that activate these kinases, and these activated kinases phosphorylate the tyrosine residue of the ITM of this Ig alpha Ig beta. So now these phosphotyrosine residues are the are actually a means of docking site for SYK kinase. Okay, so after interaction with the SYK kinase to this phosphotyrosine residue of this ITM motif of Ig alpha Ig beta, that will activate the SYK kinase, right, and that will actually transduce the signal, the downstream signal, the downstream signaling cascade will be activated. So the ITM, this is the sequence of the ITM sequence. These two Y's are conserved, tyrosines are conserved, and D or E, that indicates the aspartate or glutamate, D or E, and X indicates the random amino acid. So these tyrosines are very important in the ITM. That's why they are conserved, because they will be phosphorylated by SRC family of tyrosine kinase. This is your SRC kinase, for example FIN, BLK, LCK. They will phosphorylate the ITM and then this phosphotyrosine of ITM, that will bind with the SYK kinase, and this SYK kinase will be in turn be activated, and then it will mediate the downstream signaling cascade. So that's how the signal is transduced by B cell receptor. When antigen binds to the membrane immunoglobulin, then this Ig alpha Ig beta has ITM motif, that will be phosphorylated by SRC kinases. This phosphotyrosine will act as a platform for, or the docking site for SYK kinase. This will activate the SYK kinase, and then this SYK kinase will initiate a downstream signaling cascade, which will activate the B cell. So that's how the live B cell will be converted to the so that's how this live B cell will be converted into the activated B cell, right? So any doubt here? Sorry for the correction instability. So I will just again start with the last slide where we were. Yeah, so we saw that how the signal transduction is mediated by the BCL receptor or BCR. Right, so this is the more detailed pathway where antigen binds to the, or it rather cross links with the membrane immunoglobulin of the BCR. Then the associated Ig alpha, Ig beta, as we know that it will be phosphorylated by the SRC family kinases, the BLK, LYN or FYN. So after this phosphorylation, SYK kinase will come and it will dock to this phosphotyrosine motif of ITAM, and this will in turn actually phosphorylate the substrate which is BTK. This BTK phosphorylate, after this BTK, BLNK and PLC gamma, right? After that PLC gamma phosphorylation, PLC gamma is activated. Then this PLC gamma 2 will go to the membrane and it will digest this PIP2 to DAG and IP3, right? So this IP3 will go and it will open the pore of calcium ion channel in the ER. So the calcium ions will be in flux to the cytoplasm from the endoplasmic reticulum, and this DAG will activate the protein kinase C, and this will activate the PPCK mediated pathways, which will help to proliferation of the cells. And this GEF will also be activated. This GEF, it will undergo the small G protein pathways, which can be either in the RAS pathway or RAC pathway. RAS is a typical MAP kinase pathway. So all of them, RAS pathway, RAC, PCK mediated pathways and calcium ion channel, they will make changes in pattern of the gene expression, functional changes in the cell activation and differentiation. So that's how this signal transduction by B-cell receptor work, and they bring some changes in the pattern of gene expression, and it will help to activate the B cell, right? So that's how the naive B cell, upon the antigenic stimulation, this naive B cell upon this antigenic stimulation, they will be activated, right, by the signal transduction which is mediated by the Ig alpha beta heterodimer of BCR, which has the phosphotyrosine motif. So this is the sequence of B cell activation by TD antigen. TD means the thymus dependent antigen. So antigen crosslinks here. This red antigen crosslinks the membrane immunoglobulin, which generates the signal 1, as we saw here, that it will generate the signal 1 here. TD antigen, it will give you the signal 1. So antigen crosslinks with the membrane immunoglobulin, which will generate signal 1, which leads to increased expression of class 2 MHC and co-stimulatory B7. So it will, after this antigenic interaction, the B cell will start expressing the class 2 MHC protein and the co-stimulatory factor B7, right? This antigen antibody complexes are internalized by the endocytosis, receptor mediated endocytosis, and degraded to peptides. And these peptides, some of them will be bound with the class 2 MHC protein, and it will be go to the cell surface where this class 2 MHC will be presenting this peptide. Now this helper T cell will come and it will recognize this antigen class 2 MHC here, this one MHC class 2 antigen on B cell membrane. I think someone is in the queue. So here for thymus dependent antigens, the antigen crosslinks with the membrane immunoglobulin, which generates the signal 1, which leads to increased expression of class 2 MHC and the co-stimulatory protein B7. So now this antigen antibody complexes will be internalized by the receptor mediated endocytosis here, and it will be degraded to some peptides, and some of which will bind with the class 2 MHC and they will be exported to the cell surface, where the class 2 MHC will present the antigen here and the antigenic peptide here. So after that, this helper T cell will come and it will recognize the antigen class 2 MHC here, this complex class 2 MHC and antigen complex on the B cell membrane. This plus this co-stimulatory signal B7 that will activate the helper T cell. So this B7 will interact with CD28 of helper T cell, and these two interactions, this antigen class 2 MHC and TCR of T cell, and this B7 and CD28 of helper T cell, these two interactions will activate the helper T cell. Now helper T cell will begin the expression of CD40L. So it will express the CD40L and B cell will express the CD40, and this interaction of CD40 and CD40L, that will give you the signal 2, as we saw here. Signal 1 is this cross linking of antigen with the membrane immunoglobulin, and signal 2 is CD40 CD40L. So here, upon the antigenic stimulation, which gives you the signal 1, it will start synthesizing so it will start presenting the antigenic peptide by MHC class 2, and it will start expressing the B7 and CD28 for the helper T cell, and then after this interaction, the helper T cell will begin to express the CD40L here, right, and B cell will express the CD40. So the CD40 and CD40L interaction will give you the signal number 2. First is here antigen cross-linking with the membrane immunoglobulin, and the second is the CD40 and CD40L interaction. This will give you the signal 2. So these two signals and this B7 and CD28, this interaction will give you the co-stimulatory signal. This will give you the co-stimulation, right? After all of the signaling, the B cell will begin to express the receptor for various cytokines, right, and it will release, and the helper T cell will start releasing the cytokines. So to those cytokines which are released from the helper T cell, that will bind to the B cell, which has the receptor for those cytokines which was just recently expressed, and when B cell sense that these cytokines via their receptor of which is the receptor for these particular cytokines, so they will support the progression of the B cells to DNA synthesis and to differentiation. So it will activate the B cell, and B cells will start the will enter into the cell cycle, where it will, in the S phase, it will synthesize the DNA and it will proliferate, right, and it will be undergoing differentiation. So these signals will activate B cell. Now activated B cells will start entering into the cell cycle, which will allow the B cells to proliferate, right. So that's how the B cell activation and proliferation happens from a mature when a mature B cell when a mature B cell when a B cell gets matured in the peripheral lymphoid organ by antigen stimulation, then if it is a thymus dependent antigens, then it will give you two signals. One is the membrane immunoglobulin and the antigenic interaction here. Second is CD40 and CD40L interaction here with helper T cell, and this B7 at CD28 will act as a co-stimulation. So all of this will allow the B cell to express the receptor for various cytokines, which will be released by this helper T cell. So upon receiving those cytokines from the helper T cell, the B cell will enter into the cell cycle, where it will undergo the proliferation, right, and of course differentiation. So that is the sequence of B cell activation by thymus dependent antigen. So in case if you have any doubt, please tell me. So this is the way of activating the B cell by thymus dependent antigens. So now we'll talk about the primary and secondary response. So when antigen is infecting for the first time, then the cell will give you some elicit immune response. So first response is known as the primary response, where after after the lag phase there will be a level of IgM, and later there will be a level of IgG, which is yellow one. So first IgM, then IgG, and when the same antigen will infect the second time, then the response which will be elicited, that is secondary response. There will be a IgM initial response, and there is a IgG secondary response, which is the major one, and the secondary IgG response is more than the primary IgG response, because in the secondary response, because in the primary response will already create the memory B cell, right, which has information for the antibody which is best suited for this antigen. So in the second time, whenever the antigen it comes to the body will elicit the secondary immune response, and there is no need of learning, so because the information is already there. So that's why this level of the antibody concentration in the serum will be more, because the antibody information was there. So that is the primary and secondary in immune response. Then this is the peripheral lymph node, where this B cell go, and from bone marrow it encounters antigen, it gets activated, right, and from helper T cell and the antigen, as we discussed in the last slide, both of them can activate the B cell, and then B cell can undergo the plasma B cell or memory B cell. So this entire process is occurring in the peripheral lymphoid organ, for example lymph node. So let's see what are the components of the lymph node. So in the lymph node, which is a peripheral lymphoid organ, in in lymph node, the outer surface is cortex, is known as cortex. In the cortex, there are some primary follicle of B cell activation, there is germinal center, there is some secondary follicle, right. So in primary follicle, B cell gets activated. After activation, the B cell will go to the germinal center, where the B cell will start proliferating and differentiating. Like here, you can see that upon entering into the peripheral lymphoid organ, B cells will be activated by helper T cell and antigen. Here, this is the procedure in the lymph node. The helper T cell and antigen will activate the B cell, right. Now this activated B cell will go to the germinal center, where it will undergo proliferation and differentiation. Right. So after activating, it will go to the germinal center, where it will be proliferated and differentiation, right. Then this is your secondary follicle, and after cortex, then it will be started as paracortex, which is the actual region of initial T cell and B cell activation. And in the medulla, when B cells will go to the medulla after activation, then they will secrete the plasma cell, right. So again, in the cortex there is only B cells. So in primary follicle, B cell gets activated upon the antigenic stimulation. In germinal center, there is also a part of the cortex. The B cells will be proliferated and differentiated, and then this is the paracortex, where there is majorly T cell, where the T cells get activated. And in the medulla, this is the location of both B and T cell. So in medulla, B and T cell both and secrete uh, the plasma B cell, they will secrete the antibody. So now we will briefly see the cellular events in the germinal center, means here, where B cell, where the activated B cell comes and it will proliferate and differentiate. So now we will see how does it happen in a germinal center. First, this B cell population, they will come, right? They are known as centroblasts or activated B cell. So this, because activation occurs here after the antigenic stimulation, and of course the TH cell interaction, B cell gets activated. Then it comes to the activated B cell or centroblast. They come to the germinal center. So in the dark zone of the germinal center, that is the dark zone which allows the somatic mutation in the proliferating centroblast. So the activated B cell which are getting proliferated, they will undergo somatic mutation, which will actually generate a pool of B cell which has the different variable region in their surface immuno or the membrane immunoglobulin, right? So now some of the B cells will have have the membrane immunoglobulin or MIG in the membrane, which has the high affinity for the antigen, and some B cells will have the low affinity for the antigen. So the low affinity containing B cells will be died by apoptosis, and high affinity B cells will go to the centrocyte. So it will be known as a centrocyte, and it will go to the migrate to the light zone. So in the light zone, there is follicular dendritic cell where it will present the antigen here, right? So now when this B cells will come, then it will make a antigen antibody complex. The low affinity B cells, the black one, they will show low affinity, so they will be died by apoptosis, and the blue one which is showing the high affinity, they will be selected, because that is the way of selecting the B cells which has the membrane Ig, which has the variable region which can bind with the antigen with a very high affinity. So now this B cells which will be selected here, they will bind here, and they will be selected because they are showing high affinity. So they will migrate to they are the selected centrocyte. So they will be migrated and to the light zone, where they will interact with the helper T cell, right? And as we saw, these are the signals which will actually activate the B cell. So now this helper T cell will again activate the B cell. So B cell will be differentiated into the plasmablast and memory B cell. The plasma B cell, they will secrete the antibody, and the memory B cell, they will not secrete the antibody, but they will keep the information of the immunoglobulin gene which has the best fit with this with that particular antigen. So that's how the B cell can be differentiated to a plasma B cell and memory B cell, and that's how the B cells are selected here, which is showing the high affinity. So that's how the cellular events in a germinal center takes place. So any doubt here? So now we will discuss some questions for your assignment. That, for example, during B cell maturation, which of the following stages of the surrogate light chain complexed with heavy chain start expressing? So the answer is pre-B cell, because in the pre-B cell there were see in the pre-B cell there was surrogate light chain which is complex with the heavy chain. So the answer is the pre-B cell. Which of the following fates may occur to a B cell that reacts to a self antigen? So it can do receptor editing, it can be die by apoptosis, and it can be turned into energetic B cell. So the answer is all of the above. Interaction of the naive T cell with APC induces expression of both IL-2 and IL-2 receptor on the naive T cell. IL-2 binds with the IL-2R on the same T cell to induce their proliferation. This is an example of paracrine, autocrine, endocrine reaction, and exocrine reaction. So this is a example of autocrine reaction, because the IL-2 is secreted by the helper T cell, and that IL-2 it's bind to the IL-2R of the same T cell. So the T cell is secreting signal which is binding to the receptor of the same T cell. That is example of the autocrine function. So the answer is the autocrine. B cell receptor having high affinity for the antigen as selected in the light zone. That process is known as affinity maturation, affinity tagging, recombination of the above. The answer is affinity maturation. This will mature the B cell. It will help the B cell to select that which B cell has the highest affinity with the antigen, which is presented by the follicular dendritic cell. So this is affinity maturation. Interaction between the B cell and the TFH cell, which initiate the process of class switching of B cells, are MHC2 TCR, CD40 CD40L, both A and B, none of the above. So both are true. MHC class 2 and TCR interaction and the CD40 CD40L interaction, basically they are the two main signals which between B cell and helper T cell which initiate the process of class switching of B cell. So answer is both A and B, option C. Okay, so I think yeah, so that was all for today's session. So next week we can meet and we will discuss the next topic. So see you on the, thanks for joining, see you on the next week.